last time we discussed few of the organelles. Also we discussed the difference between prokaryotes and eukaryotic cells, as well as some of the all the parts or organelles included in prokaryotes we have discussed, then started with the eukaryotes. So some of the organelles we have already discussed. Now we'll begin with the next organelle, that is called as vacuole. Keep your textbook also with you, and as and when you find any doubt, you just note it down there itself. Okay, the vacuole is the membrane bound space found in the cytoplasm and most common to plant cells. Means when the vacuoles are present, their number is large, or even the space or the area occupied in the plant cells is more than the animal cells. So you remember that this vacuole are nothing but the membrane bound space which is found in the cytoplasm, and the concentration of the vacuole is greater than the cytoplasm because of the ions present in it. So ion concentration is increased, exchange of ions takes place in the vacuole. So their concentration of the vacuoles, because of the different types of ions present in it, is more than the cytoplasm. Now in the cytoplasm itself, different types of organelles are present. So vacuoles are one of the next, that means organelle which is considered to be important for specific functions. Now what are the functions that we'll discuss step by step? In some plants, vacuolar sap also contains some pigments like anthocyanin, which impart color to the plant part. Now, anthocyanin is one of the examples of the pigments. Its color is blue. Like that, different types of pigments are present in the vacuolar region. Sap is the content, that concentration, liquid part present in the vacuole. So in the vacuolar sap, that means in the vacuole itself, different types of pigments are present which give color to that particular plant part. For example, in case of petals, flowers, different colored flowers, that color of the flower is because of the petals. And what the petals contain, different types of pigments, and these pigments are present maximum in the vacuolar region, vacuolar sap. Okay, so vacuoles, they're important. One of the important roles is that they show the presence of pigments. That means they storage is the main purpose. Like that pigment, few other important uh, fats, oils, these compounds are also stored in the vacuole. So storage is main important function of the vacuole. So one of that is what? And pigments are present, and some of the pigments, as per the type of pigments they store, it gives the color to the plant part. For example, different colored flowers, that means the petals, they show different colors because of the pigments present in the vacuolar region. Now in plant cells, the vacuoles can occupy up to 90% of the volume of the cell and play important role in osmoregulation. Osmoregulation, osmotic pressure is maintained. So that pressure is regulated, and that is what called as osmoregulation. And this is very common in case of prokaryotes also, and some of the other organelles, so many aquatic organelles, there also they are playing very important role. We'll come to that part. Now there are different types of vacuoles. For example, gas vacuoles, contractile vacuoles, food vacuoles, and as per the name, they are playing its role. Okay, but the main important function of the vacuole is osmoregulation. Osmotic pressure of the cell with the surrounding is maintained with the help of these vacuoles, with the help of exchange of ions present inside the vacuole. So exchange of ions takes place, concentration gradient of the ions is increased inside the vacuoles, and that's why its concentration is more than the cytoplasm. So this is what, which is related to osmoregulation also. So osmoregulation is what? Osmotic pressure is maintained, and not only in case of plants, in case of animals also, this, and especially aquatic animals, this is very important role. Now in plants, the tonoplast facilitates the transport of number of ions and other materials against concentration gradients into the vacuole, and hence their concentration is significantly higher in vacuole than the cytoplasm. That's what I told just now. Tonoplast is nothing but the vacuoles, the layer, outermost layer of the vacuole, which is a single layer. That is what called as tonoplast, and that facilitates the transport of ions. Which type of ions are to be taken inside the vacuole? Then the concentration of which ions is to be increased, which ions are to be released out of the vacuoles, that is maintained by tonoplast. That is the layer of the vacuole, and accordingly their concentration, ion concentration, is more in vacuoles as they are storing, storage purpose these are used mainly for. So their concentration is significantly higher than the cytoplasm, which is outside. Now these three types of vacuoles are playing important role: gas vacuoles, contractile vacuoles, food vacuoles, and the main function is what? Generally osmoregulation, but along with that additional function is also done. Now in case of amoeba, the contractile vacuole is important for excretion. Now you must have learned the structure of amoeba also. Amoeba, what is the main important or specific uh, role of amoeba? I can't say the role, but what is the specificity of amoeba? That it shows the presence of pseudopodia. Pseudo means false. Podium means legs. So false legs like structures are present, and it has no specific space, shape, right? So as per the requirement, it changes its shape. Now what happens? It shows two types of vacuoles. One is what? Contractile vacuole, which maintains the osmotic pressure of the cell with the surrounding, and the another one is what? Food vacuole. Now what happens? Food vacuole, that means any foreign material or the food. In case of amoeba, suppose bacteria is the food, when it comes in contact with it, this organism, it increases their organs like the pseudopodia and engulf that material, foreign material, as its food, and then it is surrounded by the pseudopodia, and then it works as a food vacuole, where then different types of enzymes are released which are helpful for the digestion of that particular food which is taken inside. Now, in many cells, as in protist, food vacuoles are formed by engulfing the food particles. Just what I was giving you the example of amoeba. In case of Paramecium also, if you see, there are two types of vacuoles. One is food vacuole and the other is contractile vacuole. So, food vacuole and contractile vacuoles, little difference is there. Food vacuoles is what? For engulfing the foreign material as a food, and then that releasing the digestive enzymes, and that is working, that vacuole is working as a food vacuole, where the different types of enzymes, that means digestive enzymes, are released there, and then food gets digested over there. And the other one, contractile vacuole, is generally maintained, is used for maintaining the osmotic pressure of the cell itself, body itself, with the surrounding. Okay. So these are the two examples I mentioned you in case of food vacuoles and contractile vacuoles as per their functions. Now this is what one more example, the same example of amoeba, which is shown in with the help of diagrams here, pictures here. See, this is amoeba, irregular shaped body of amoeba having the nucleus, and these are called as pseudopodia, that means false legs like structures. Now when the food material comes in contact with the amoeba, like bacteria, suppose here this is the material, food material coming in contact with this.
Ma'am, your voice is not here.
These pseudopodia, they increase and they form a layer, or they false layer surround the material, that is the food material, and this is what in the second diagram you can see this food vacuole has formed. Now there, and digestive enzymes are released. That means this pseudopodia enclose the food particle which comes in contact with this, and then these enzymes help in the digestion of this food material. And then once this food gets digested, the remaining part, which is called as excess of undigested part, or excess of part, that is called as excreta, which is undigested, that is released out again. The pseudopodia, with the help of pseudopodia itself, that undigested part, that is called as excreta, is released out from the body. So this is what, for ingestion of the food as well as for the excretion, these pseudopodia are playing the role. And the main important point what we are discussing here is the food vacuole and contractile vacuole. That is the role, important role of in case of these organisms. Now the next important organelle everybody knows, and that is what? Mitochondria. You have learned this structure in your school days, right? But in a small, or you can say in a very limited knowledge. Now, in detail, we will be studying the structure of mitochondria. Mitochondrion is singular, mitochondria is plural. Okay, this is very, very important organelle present in all type of eukaryotic cells, all type of living organisms, I can say, prokaryotes as well as eukaryotes, but their function in prokaryotes is different. Now we will be talking about the eukaryotes. In eukaryotes, mitochondria, they are playing very important role. Unless specifically stained, they cannot be seen by simple, that means the compound microscope. Under microscope, they can't be seen. So they are to be stained. Then only they can be seen under microscope, that they are visible. Now, about the shape, size, number, it varies from cell to cell. The number of mitochondria per cell is variable depending on the physiological activities of the cells. Now, as I said, the number of mitochondria, the shape of the mitochondria, the size of the mitochondria, it varies from cell to cell. So, as per the requirement, it is seen. Typically, it is sausage shaped or cylindrical, it is spherical, sometimes oval, rounded. So, there are different shapes seen in case of mitochondria. And diameter also ranges from 0.2 to 1 micrometer. Average is 0.5 micrometer. And the length also varies from 1 to 4.1 micrometer. So this is what variations seen in shape, size, as well as number in case of mitochondria in different types of organelles. Each mitochondrium is a double membrane bound structure with the outer compartment and inner compartments divided by lumen. Lumen is the space. Lumen is nothing but a space in between. So when you see, this is the structure of mitochondria in detail. Now we'll see, this is in your textbook very clearly shown. This is the outer layer, which is smooth, whereas inner layer is folded. You can see these folds on the inner layer, and there is a space between the outer and inner layer of mitochondria, and that is called as intermembrane space. That lumen means nothing but intermembrane space. Now, if you see the inner membrane, that inner membrane, it show inside the inner membrane there is a matrix, and that matrix shows different types of organs. What are these different materials present in? Circular DNA in the matrix. Ribosomes are present. In case of ribosomes, specific type of ribosomes are present here in mitochondria, small. Then along with that, different types of proteins and very specific structure which is seen on the inner layer of mitochondria, that is called as oxiosomes. Now this is the folding, one fold of the inner membrane of the mitochondrium. This is the single fold, and this single fold, this is the outer membrane, this is the inner membrane, and on the inner membrane you can see the lipid layer as well as protein layer. And if you see this structure, darkly stained structure, that is called as F1 particle. They are also called as oxiosomes, okay, or they are also called as electron carriers. And this F1 particle, actually it contains oxyosome that contains three parts. This part is called as head, that is oval in shape. This is the stalk or pedicel, and this is the base. So oxyosome shows three parts: head called as F1 particle, stalk or the pedicel, that is called as F0 particle, and the base, which is embedded in the inner membrane. And these heads generally are meant for carrying, these are also called as electron carriers. These electron carriers are transported from one oxyosome to the another. Now the actual role, when we will study the respiration part, that time you will come to know, but just remember that this is the structure of oxyosome. This is the section of mitochondria which shows the outer membrane which is smooth, inner membrane with the folds. there is a space between the outer and inner membrane that is called as intermembrane space or lumen. Inside the inner membrane, the matrix is present, which shows the presence of circular type of DNA, ribosomes, some enzymes and proteins. And the most important part which is present, that is called as oxiosomes, and the head is towards the matrix and base is embedded in the inner membrane. So this head is in the matrix, whereas this base is embedded in the inner membrane. Remember that. Sometimes the students may make mistakes while drawing it. I will tell you again. So what is the main role of this? That is respiration. Mitochondria play a very important role in respiration. They are the sites of aerobic respiration. Now, as I told you just now, that in case of mitochondria, whatever these folds, they are called as crista. Here, each fold that is called as crista, and the plural is cristae. So outer membrane is smooth, but inner membrane is showing the folds. They are called as cristae. Okay, and each crista shows the presence of number of oxiosomes with these three parts, and their role is electron carrier or electron transportation. Mitochondria produce cellular energy in the form of ATP. Hence, they are called powerhouses of the cell. You must have heard this, and you have learned also in the previous standards that give reason. Mitochondria are called powerhouse of the cell. Why? Because they produce energy, and that is stored in the form of ATP. And as and when required, that is used. That ATP is used. That means the energy is stored in mitochondria, which is produced through cellular respiration, and that is in the form of ATP, that is adenosine triphosphate. Mitochondria matrix, what it shows, as I mentioned just now, that is circular DNA. Then ribosomes, as I said, small ribosomes, that means 70S type of ribosomes, and few RNA molecules. So, and this is what the mitochondrial matrix shows. Important or parts present in the these chemicals, or you can say these material present in the matrix are single circular DNA molecules, 70S type of ribosomes, and few RNA molecules. So the mitochondrial matrix contains these parts. So I hope you have understood this part at least. I will repeat this part again. In case of mitochondria, outer membrane, inner membrane. Outer membrane is smooth, inner membrane is folded. These folds are called as cristae. Each crista shows, this is a singular crista in this diagram shown. It shows the presence of oxiosomes on it. These are oxiosomes. This is inner membrane, this is outer membrane. These are the oxiosomes. They are also called as electron carriers. They are also called as F1 particles. And if you see this single structure of oxyosome which shows the head oval, that is called as F1 particle or F1 part. Base, that is this which is embedded in the inner membrane of the crista, and the stalk or pedicel which connects head to the base. So this is called as pedicel or F0 particle. So head is towards in the matrix, head is present in the matrix, and base is embedded in the inner membrane. So remember this. So this is the structure of mitochondria, and the role, main important role is what? Cellular respiration, and by cellular respiration, whatever the energy is produced, that is stored in the form of ATP, and that's why it is called as mitochondria are called powerhouse of the cell. So mitochondrion is called as powerhouse of the cell, which is very important for give reason it can be asked. Now the next is plastid. The next organelle is called, there are different types of plastids present. They are present in, these plastids are present only in plant cells and euglenoids. So you remember in euglena, the photosynthetic pigments are present, so that's why they are called as plastids. But the major And the most important part, that is, you can say, in case of plants, only plants only plastids are present. They cannot be seen in the animal cells. Okay, they are having different colors, and as per the color, that means the color of the pigment present in it, they are known by different names. Now they bear specific pigments, and according to the color of the pigment, they are known as chloroplast, which are green in color. If the pigments are green in color, they are called as chloroplast. Different colored pigments are called as chromoplast, and without color, when colorless, they are called as leucoplasts. So I'll repeat, depending on the type of pigment color, they are known as chloroplast when green, chromoplast, colorful pigments, leucoplast without color pigments. That means the pigments, they are considered to be colorless. Now, step by step, one by one, we will discuss all these different types of pigments. Chloroplasts contain chlorophyll and carotenoid pigments. Chlorophyll and carotenoids are responsible for trapping light energy essential for photosynthesis. So not only the color as green, but as you know that chloroplast, the main function of the chloroplast is what? As they are green in color, they are present in all green parts of the plant. And what is their main function? Everybody knows that is photosynthesis. And that is because of what? The pigment which they contain, that is called as chlorophyll, as well as carotenoids. Along with green colored chlorophyll pigments, carotenoids, these are yellow and orange color pigments. They are also present in that, and they are responsible for trapping light energy. That means this light energy from the solar, that is the solar energy, light energy which is received from sun, that is trapped with the help of these pigments for the process of photosynthesis. So trapping of the solar energy is important role of chlorophyll pigments. Now this is the structure, detailed structure under electron microscope. You can see the structure, and everybody remember the role of each part present in the chloroplast. This chloroplast is also double membrane structure, outer membrane and inner membrane. So both the membranes are smooth here in case of chloroplast, outer as well as inner membrane. There is a space between these two membranes, outer and inner membrane, that is called as intermembrane space. Inside the inner membrane, you can see these green colored structures which look like stack of coins. The coins are placed one above the another. Like that, you can see that structure, the entire pile of these stacks like coins of stack of coin like structures, the entire structure is called as granum. So these all stacks or these green colored structures which are looking like a stack of coins, these coins are placed one above the other, that is called as granum. This is singular granum, this is another granum, these are all grana. Okay, and each granum, it shows the presence of disc-like structure, oval disc-like structure, that is called as thylakoid. So these thylakoids are placed one above the other. These thylakoids are placed one above the other, which gives the appearance of stack of coins. Right? Now in the next diagram, we will see again the structure where here, if you see the structure, each thylakoid, there is a space, you can see one more thing, that these grana are connected to each other by these tubular structures. Can you see here? These are called as intergranal lamellae. These are called as intergranal lamellae, or also called as stroma lamellae, which are connecting to neighboring grana. This is the single granum. This is also single granum. Here are the grana. All these grana are connected to each other by tubular structure. That space, that is called as stroma lamellae or intergranal lamellae. Okay, so this is Now, as you know, these thylakoids, each thylakoid, now again, the number of the thylakoids in each granum also varies, and the number of grana in each cell also varies. And shape generally is as, okay, generally these are oval shapes, sometimes they are spherical shapes, some are circular shape, sometimes they are even ribbon shaped. Okay, so there are their size varies, their number also varies, and the structure is same, internal structure is same. That is, they show outer layer, inner layer, there is a space between the outer and inner layer, that is called as intermembrane space. Then the inside the inner membrane, you can see the matrix. That matrix show, that is called as stroma. This matrix is called as that semi-fluid structure. That matrix is called as stroma, which is responsible for the respiration, whereas these grana, as they show thylakoids, and the pigments present in it, photons, they are different colored pigments present in it. They are responsible for trapping the solar energy, that is the light energy, and converting it into the chemical energy. So these grana, stack of coin-like structures, they are called as thylakoids, present one above the other. They are connected to each other by stroma lamellae. So this is the structure of chloroplast. Outer membrane, inner membrane, inside which the matrix which is present called as stroma, that is responsible for chemical reaction, whereas these grana are responsible for light reaction of the photosynthesis. So main function of the grana is what? In case of chloroplast, that is the photosynthesis, because of the pigments present in it. Now, the fat soluble carotenoids. Carotenoids, these are carotenes, xanthophylls. Carotene, orange colored pigments, xanthophyll, yellow colored pigments, and few more others are present. So together they are called as carotenoids. Carotenoids contain carotene, orange color pigments, xanthophyll, yellow colored pigments. So together they are called as carotenoids. They give yellow, orange, red colors to the plant parts. So different parts of the plant, especially petals, because of these pigments, the color flowers are colorful, showing their colors. Then one more different example of these plants, with the help of pigments present, accordingly they give the color. Now the third category of the plastids, that is called as leucoplasts. Leucoplasts, they are colorless plastids, and also they vary in shape as well as size, and depending on the type of nutrient they store or the nutrient which is present, they are known by different names. So leucoplasts are colorless pigments. They are colorless plastids, but their number also varies, their size varies, their shape varies. So varied shapes and sizes with the type of nutrient stored in it, they are known as amyloplasts, elioplast, alluroplast. Now you can very easily remember their names. Now amyloplasts, which shows the presence of amylose, that is the carbohydrates as well as starch. Now here in case of potato, this is the example which shows the presence of leucoplast and sometimes chloroplast. So when the green colored pigments are present, that is called as chloroplast, and colorless pigments they called as leucoplast. This is just example. Now see, amyloplast store carbohydrates. You know, amylase. Have you heard this word? Amylase, that means the enzyme which digest amylase, that is what? Starch. So amyloplast, they store carbohydrates in the form of starch. So they store carbohydrates, and the example is potato. Everybody knows Potato store starch. So in potato, which type of plastids are present? Leucoplast, and in the leucoplast, amyloplast, that is which store carbohydrates, that is starch. Then Aleoplasts means they store oils and fats. Aleoplasts are the plastids. The name aleoplast is because of the presence of store material that is in the form of oils and fats. So aleoplasts are the plastids which store oils and fats. Example is seeds of castor. You know, castor seed oil. Castor oil, you must have heard the word. So seeds of the castor, they show the presence of oil, that is in the fats and oils, and so the plastids are of aleoplasts. Plastids, that means pigments which are showing the presence. These are colorful, colorless like pigments, but they are storing oils and fats. The third category of leucoplast, which is called as alluroplasts. Now, if you remember the word alluron, which is a type of protein. Okay, so alluroplasts are the plastids which store proteins. And the examples, you know that all the pulses, the seeds of all pulses, which produce oil, which you know everybody knows different types of pulses, the examples from which we get the oil. So all these pulses, seeds of the pulses, they show the leucoplast called as alluroplast, that is a type of plastids, colorless plastid, and the main function of this is the storage of proteins. So all the pulses which are considered to be important for protein gradient, that means the proteins are more in the pulses, that is because of these alluroplasts. Okay, and all the different types of seeds from which we get oil, most of the seeds from which we get the oil, but the example which is given, castor oil, very common example, these, that is the plast, alluroplast, and amyloplast, the common example, potato. So these amyloplast, alluroplast, and alluroplast, these are the colorless leucoplast, and as per the type of storage material present in them, they are known by. Okay, majority of the chloroplasts of the green plants are found in mesophyll cells of the leaves. If you have learned the structure of leaf in your previous standards, you might be knowing that the structure of leaf, the major part of the cells, that is mesophyll cells, they are lens shaped, oval, spherical, discoid, or even ribbon like organelles. Means these mesophyll cells shows the presence of green color pigment. Okay, so mesophyll cells, if you see in case of leaves, all green part, in case of plants, that is showing the presence of chlorophyll. Now again, one more structure, you can see this diagram. Here the outer membrane, this is the inner membrane. There is a space between present between the inner and outer membrane, intermembrane space. This is the 3D diagram of chloroplast, and the length varies, width also varies. That range is 5 to 10 micrometer, width range is 2 to 4 micrometer, and their number also varies from 1 per cell of the Chlamydomonas, that is a green alga, to 20 to 40 per cell in mesophyll. That means Chlamydomonas is a cell, that is alga, a type of green alga which belongs to chlorophyceae, and this Chlamydomonas, that shows the presence of chlorophyll, and its number is only 1. Means this is the example where only 1 chlorophyll is present. But in case of mesophyll cells in plants, 20 to 40 such chloroplasts are present. So that is why this is just range given, that its number varies, shape also varies, length and width also there is a range, but varies from cell to cell. Just like mitochondria in chloroplast also, the size, shape and number varies from cell to cell. Now, this is the one more, especially again, it is distributed all the parts in this diagram. This is simpler 2D. we studied just now, you observed 3D diagrams. Now this is the very simplest form of chloroplast, where these two membrane structures you can see, outer membrane, inner membrane, and this single granum, this is one more, another granum, this is another granum, and the matrix called as stroma. The matrix which is present inside the inner membrane, that is semicellular solid structure or fluid structure, that is called as stroma, and singular is granum, plural that means these many grana, they are called as grana, singular is granum, and each granum, as I told you, that shows the presence of thylakoids present one above the another. That is, these thylakoids are placed one above the other, forming or showing or giving the appearance as that of the stacks of coins. This is the simpler structure, and again in this structure you can see the section of this chloroplast, where these, when you see the internal structure, these are the lumens, that is the space between the two, that is the space between the membrane, that is called as lumen. Okay, this is the singular granum, and this granum is connected to this granum by this tubular structure, that is called as intergranular lamellae, or this is also called as stroma lamellae. So very simple form. Now I have repeated 4 times, I think more than that also, so that you must have learned now the structure of this granum, the structure of chloroplast. So flat membranous tubules called the stroma lamellae connecting the thylakoids of different grana. So as I showed you in this diagram also, that this tubular structure is called as stroma lamellae, which contains the space which is connecting the two neighboring grana. Membrane of thylakoids encloses a space called lumen. Now this is single thylakoid, this is another thylakoid. Each thylakoid is placed one above the other, and the space between the membrane which is inside the membrane, that is called as lumen. So this is one more structure, again 3D structure of the chloroplast you can see. So the stroma contains enzyme, and pigments are responsible for trapping light energy for photosynthesis. So each and every part of the chloroplast is very important. The membrane, the stroma, which contains the enzyme for respiration, the dark reaction, photosynthesis, which shows 2 parts. In the next chapter we will be discussing that again, light reaction and dark reaction. So grana are responsible for the light reaction of photosynthesis, and where their main important part is what, these pigments, they are trapping the solar energy required for the photosynthesis. So this is what about the chloroplast. Now about the next organelle called as ribosomes. Next organelle is what? Ribosomes. Ribosomes are again of different types, but here in case of chloroplast, again you can remember chloroplastic ribosomes are small, that means like prokaryotic 70S type of ribosomes, whereas cytoplasmic ribosomes in case of eukaryotes, they are 80S type. Like in mitochondria we discussed that whatever the matrix present, it shows the ribosomes of 70S type. Same here in case of chloroplast also, chloroplastic ribosomes are small, that is prokaryotic type, that is 70S. But the cytoplasmic ribosomes in a single cell, eukaryotic cell, in the cytoplasm, whatever the ribosomes are present, they are of 80S type. So ribosomes of the chloroplast are smaller than cytoplasmic ribosomes. Now in detailed structure of ribosomes, ribosomes are granular structures first observed by George Plastids in 1953, and they are composed of RNA and proteins. What important thing you need to remember is that they are of 2 types. Eukaryotic type of cells show 80S type of ribosomes, whereas prokaryotic type of cells show 70S type of ribosomes. Now what is 70S and what is 80S, I will tell you. S, that stands for sedimentation coefficient, expressed in Swedberg's unit, and it is indirectly a measure of density and size. Now, if you see, this is the diagram which is showing the presence of ribosomes. Okay, this is a single ribosome, and these are forming a chain-like structures. They are called as polysomes. When ribosomes are in a chain, you can see they are called as polysomes. And S, why we are using 70S and 80S. S stands for sedimentation coefficient. That that is called as Swedberg's unit. That represents the density and size, that measures the density and size of the ribosomes. So 70S and 80S, what we call. Now, how we consider 70S and how we consider it as 80S. 70S, there each ribosome, if you see, that is composed of larger and smaller subunits. Each, now in the previous diagram also I can show you, this is the ribosome where this is a smaller subunit, this is a larger subunit. Everywhere you can see one is smaller, one is larger. Here they are separated. During the process of protein synthesis only, they come close to each other. Here, smaller subunit, larger subunit. So in this way, these two subunits, they join, and there, as per their density, see, S, that is Swedberg's unit. 70S ribosomes are made up of three subunits. Larger subunit is of 50S and smaller subunit is of 30S. But their total doesn't come 80. Here, they are separate. But during the process of protein synthesis, when they come close to each other, then their role, then they are playing the role of protein synthesis. They become 70S in prokaryotes, and in case of 80S, these subunits are of 60S and 40S. Larger subunit is of 60S. Smaller subunit is of 40S type in case of 80S type of ribosomes present in eukaryotes. Now these are the subunits. See, you can see here larger subunit in case of 70S type of ribosomes, 50 Swedbergs. Here it is 30. These are separate. When they join, it becomes complete. Then you can see when they come in contact with each other during the process of protein synthesis only, and they form 70S. In case of eukaryotes, larger subunit is of 60S, smaller subunit is of 40S, and then when they join, the complete one, only during the process of protein synthesis, they come in contact with each other and they become 80S. Their total doesn't become 100, its total doesn't become 80. Remember that 50 and 30 are separate. When they are separate, when they are complete during the protein synthesis, their gradient is 70S only. Same in case of eukaryotes. When they are separate, they are of 60 as larger and 40S as smaller. But when they come in contact with each other, it becomes 80S during the process of protein synthesis only. So this is the structure of eukaryotic ribosome in large and small subunit. And the two subunit in both types of ribosomes associated with each other in presence of magnesium ions. So only in presence of magnesium ions, these two subunit they associate with each other, and they provide space as well as enzymes for the synthesis of proteins. So this is what we discussed about these organelles. I will repeat which organelles we discussed. That is ribosomes, then different types of plastids, chloroplast, chromoplast, leucoplast, then different types of leucoplasts. Then after the plastids, before that, we discussed, wait a minute. So after plastids, we discussed mitochondria, its structure, total structure, then vacuoles, different types of vacuoles as contractile food vacuoles, etc. So different types of vacuoles we discussed. So how many organelles we discussed? I will stop right now. We will be having discussion about this now?
Madam, are you there? Kotwal madam? Kotwal madam. Students, please wait. I'll just check. There's some technical problem, I think. Wait. Voice, I can't hear you, madam.
Yes, Maddy, and
Voice is breaking.
present also. My net, because of power cut
Okay.
at my place, but I have changed to this one. I have changed the mode. I have taken the hotspot. So I have stopped my presentation. Now I'll stop the recording also.
Okay. Students, are you there? You